In the chapter Human Health and Disease, we have the topic on HIV/AIDS. Today, in this video lecture, we will try to learn about HIV/AIDS, the symptoms, the causal agent, then how to prevent it, and mainly the life cycle of HIV. So let us start with the discussion. AIDS. The full form of AIDS, acquired immunodeficiency syndrome. Now, acquired, why is it called acquired? Because this particular disease mainly affects our immune system, which we acquire during our lifetime. As we have already learned about the immune system, that is, we have two main types of immune system, innate and acquired. Innate is the one with which we are born with, and acquired immunity is the one with which we have acquired, which we have built in us during our lifetime after birth. So in this particular type of disease, that immune system is affected. Immune, definitely the word acquired immuno, that is the body's defense system is affected. Deficiency, that is not working properly. Syndrome. Now syndrome means a group of signs and symptoms. That is, when a person is suffering from AIDS, the disease AIDS, the particular person will not be characterized by one particular or two particular symptom. The symptoms can be a group of them. That is, as the immune system is affected, so what happens is the particular patient may be infected by a number of pathogens. His immunity is not working so as to evade out the germs, so as to kill the germs. So as a result, what happens? He can be infected by numerous antigens, numerous infections. As his entire immune system has come to a halt. It's not working to kill away the germs. So the signs and symptoms of AIDS, mainly of course the malfunctioning acquired immune system, but due to that the symptoms will be a group of them, different signs and symptoms which we usually see when we are infected by some pathogens. Now, the same, it is explained here, transmitted from person to person. The AIDS is a highly contagious disease. It affects the immune system, as I already mentioned, malfunctioning of the immune system, and they may experience a wide range of diseases. Causal organism, HIV. What is HIV? HIV is human immunodeficiency virus. Because it infects humans, so the term human is applied here. Immunodeficiency, that means this particular virus affects our immune system. That is why immunodeficiency, the virus. Now the HIV, it belongs to a special category of viruses known as retrovirus. Retroviruses, like any other virus, they have the genetic material enclosed by a protein covering, capsid or capsomeres. Now this protein covering, the capsid or the capsomere, encloses the genetic material. As we know, viruses can have the genetic material either DNA or RNA, but this category of virus, they have RNA as the genetic material. The specialty here is other viruses can also be there with RNA, but the specialty in this type is that they have a special type of enzyme, reverse transcriptase. They have a type of enzyme which is known as reverse transcriptase associated with the RNA. Now this reverse transcriptase, what can it do? It can carry out reverse transcription. As we have learned about transcription means formation of RNA from DNA. So DNA to RNA is transcription. But if it takes place in the reverse manner, that is RNA to DNA, we will call it as reverse transcription. And that is possible only in the presence of the enzyme reverse transcriptase. And this enzyme is present in these type of viruses or some viruses. Those viruses we call as retroviruses. So the unique property of retrovirus is the presence of reverse transcriptase. So, and this particular type of retrovirus, that is HIV, is the one which infects humans to cause AIDS. Now let us see in the next coming slides, we will see the life cycle, how it does so. Now the major signs and symptoms, mainly weight loss, diarrhea, fever. So these are some of the major signs and symptoms which are actually due to infection by other germs, say Mycobacterium, Toxoplasma. So these type of germs, when they infect the body, then symptoms can be varied types, different types. Some minor symptoms, the lymph node enlargement. Now lymph node is the structure when we were doing about the immune organs. Okay, the organs involved with immunity. Lymph node is one such which stores lymphocytes. So here it's affecting lymphocytes. So here you can see the enlargement of it. Then the pharynx gets infected. Different types of infections, minute infections which can otherwise mean nothing. You get cured in 1 or 2 days. But here, if this particular person who is infected with HIV will not cured by it, and it can take a drastic turn. Now, what are the modes of infection? The main mode of infection of HIV is through fluid, body fluids. Body fluids which can be blood, it can be lymph or some secretions. So the main mode of transmission of HIV is through body fluid. So whenever any two persons, a person who is having HIV, who is having HIV in his or her body, the body fluid, his or her body fluid comes in contact with another person's body fluid directly into the body, then it's likely that the second person may be infected with HIV. Now, how can that transmission occur of body fluids? That can happen through sexual contact. Sexual contact, that is through when there is exchange of the fluids, like say semen, vaginal fluids. So through that, even HIV can get transmitted from one person to another. Then through injections. Now injections means when we are taking any injections, any form of injections, through the syringe, the needle of the syringe. If the same needle, same syringe is used by many persons, it is shared, then it's likely that some fragments, some particles of blood maybe or tissue fluid may be attached to it, stick to it, and so thereby what will happen, the tissue fluid will again enter into the second person that may be a carrier of HIV. It's more often seen in case of drug abuse, drug addicts, because if they are taking they consume drugs, that means they're taking drugs through injections, then they are likely to share the syringes. So in them this chances increases. Then a small child may be infected through a infected pregnant mother. If the mother has AIDS, and is likely she may pass it on to her child who is developing inside her body. Then occupational exposure is always there. Doctors and nurses, if they come in contact with a AIDS patient, so somehow they come in contact with the body fluid of that infected person, so they can also be getting it. Then the maximum chances again occurs in case of blood transfusion and organ transplanted. Some diseases like say thalassemia, sickle cell anemia, they require regular blood transfusion. So if somehow they get blood from an infected person with HIV, then they will also be getting infected. So it is very much important that the blood has to be tested before transfusion. Same way in case of organ transplants. So these are the main modes of transmission of HIV. What the point to remember is that HIV can be transmitted only through body fluids. So if we avoid the contact of body fluids directly with our body fluid, then we can always prevent ourselves from getting the disease. Now, some points to discuss regarding infection. Now, whenever HIV enters into a human, into the person's body fluid, say blood, then it will be carried by blood. Blood contains macrophages. Now, the first macrophages are immune cells. Now, they will first infect the macrophages, the virus particles. The virus particles, their normal mode of replication Only the genetic material enters. As they have reverse transcriptase, they will carry out reverse transcription, thereby form a DNA from its RNA. The DNA will then further undergo replication, normal mode of replication into the human, and then it will help in replication of the virus. The virus comes out of that cell and further it will go and infect the T helper cells. T helper cells means the T lymphocytes, a type of T lymphocytes, that is T helper cells. It will go and infect. That will lead to decrease in the T lymphocytes. When there is a decrease in T lymphocytes, that means it kills the T lymphocytes. When there is decrease, then the chances of infections, different types of infections, as simple as common cold, as simple as common flu increases in that particular person. So we will say that the person is showing symptoms of AIDS. Now, a thing remains here, that is, a person can be HIV positive but may not be having AIDS. A person may die as being HIV positive but never show the symptoms. That is, if suppose the virus remains confined to the macrophages and somehow it could not come out to infect the helper cells, then the person will be carrying the virus but will not show the symptoms. So, since the time of infection until the expression of the symptoms, a time lag is there. A time lag is there which can vary from few months to many years even. Now, coming to the test, how can we detect that a person is suffering from HIV AIDS? The main test is ELISA. It is called as ELISA test, enzyme-linked immunosorbent assay. Enzyme-linked immunosorbent assay, in this antigen antibody, it is based on the principle of antigen antibody testing. Then how can we treat AIDS? Basically, till now there is no treatment for AIDS, but of course the longevity of the person, of the patient, can be increased by giving antiretroviral drugs. Antiretroviral drugs, that is, it will act against the virus to decrease its replication. It will decrease to some extent, but it cannot be killed. So the best thing is prevention. Preventing oneself from coming in contact with any tissue fluid from any person, because we do not know in whom the virus is growing, in whom the virus is replicating, we do not know. So we should always prevent from exposing ourselves to any body fluids without prior testing. Then very important is creating awareness, generating awareness among the public about the disease and about its spread, to educate the people and to help in these awareness programs and educating the people. An organization, NACO, that is National AIDS Control Organization, they are playing a very big lead role in spreading awareness about AIDS. Now we will come to the life cycle of HIV. Now, as soon as the virus infects the body, here is the HIV. As soon as the HIV infects the body, it attaches itself upon the immune cell. Here it is T cell has been shown, but first of course it will go and infect the macrophage. So it attaches upon the macrophage, the membrane of the macrophage, and as like a normal virus, any other virus, it will first let its genetic material enter into the cell. So its genetic material, that is RNA. Here's the RNA, it will enter. So the first step is attachment upon and the membrane fusion, followed by the transfer of the genetic material RNA. The RNA will then undergo reverse transcription. Reverse transcription being the second step, that is, with the help of reverse transcriptase, the RNA will form the DNA. Now, what DNA is this? This is the viral DNA. That viral DNA, it is double-stranded. So, what will happen? That viral DNA will get linked with the human host cell DNA. It will get linked with the cell DNA, human DNA. It will get linked. As you can see here, it is getting linked here. Integration. We call this as the third step, integration. Now normally the human DNA, all the events, it will go on, it will replicate DNA replication, so its number will double. As it doubles, what is doubling along with it? The DNA of the virus will also replicate, that will also become double. Then gradually what will happen? The DNA, the human DNA, it undergoes transcription. So as it undergoes transcription, even the viral DNA is undergoing transcription. So it goes on in that manner. So here you can see the green part, which is shown as the DNA, the part of the virus. This is the viral part and these are the human part. So what will happen gradually? So all of them, they will go on but undergo transcription. RNA, after transcription, as we know, it moves out to the cytoplasm and it undergoes translation. Now the translation process of the RNA coming out of the DNA, it involves both human translation, human protein formation, as well as viral protein formation. So what will happen? Even viral protein will form. Now that viral protein, what will happen? It will again go to, those are viral protein, that means they have the ability to form the capsid, the capsomere, the covering of the virus particles. So they will form the covering of the virus particles, and already the viral RNA is already present. So the viral RNA will now get enclosed again within the protein coat, and finally what will happen? They will bud out. They will erupt out of the cells, the macrophages. They will come out of the macrophages, thereby increasing the number. The number, if one had entered, while coming out there can be many. In hundreds they can come out. So they will be released out, which we call as viral budding. So this is called as budding. The viral particles have come out. They will again further go to infect other macrophages or the T cells. If they continue with the macrophages, they are not infecting T cells, then maybe the symptoms will be lesser. We will not see the symptoms. Of course, macrophages will be affected in that manner, but the symptoms will not be seen. But as soon as they go on attacking, they start attacking the T cells, symptoms will be seen in that particular person. As macrophages are mainly responsible for multiplying the viral particles, multiplying the HIV, they are also called as HIV factories. So the life cycle of HIV we can sum up in 7 steps: attachment, reverse transcription, integration, transcription, translation, assembly, and budding, the release of the viral particles. One more slide here, which is in a very compressed manner, it has been shown here. Here you can see the viral particle, it binds, it fuses, releases the genetic material with the reverse transcriptase. The reverse transcription occurs, it joins with the DNA, which will replicate, also produce the RNA. The RNA will then go to help, it will carry a translation. Translation will release the proteins, viral proteins. For humans also it will release, but the human part, it will form the viral coat, and finally the virus particles will be released out to infect more of the cells. Now as the viral DNA formed due to reverse transcription has become a part of the human DNA, so what happens? It will never go out. It will help in production of new viruses, but it will never move out because it has already become a part of the DNA of human. It has become a part. Unless and until that particular cell dies, the DNA will also, the viral DNA also will remain to be a part of the human cell. So that is why it is said to be an incurable disease. That is why we say that it cannot be cured, because it becomes a part of the human cell. So that is about HIV/AIDS, the basic concepts which we need to know. Furthermore concepts are there, but at the level of class 12, these concepts are sufficient for us to understand. Thank you very much. If you have understood properly, if you have followed the lecture properly, then do share the video and do press the like button. Thank you.